Nitrosamines are a class of carcinogenic chemicals typically found as impurities that represent a major health risk to patients due to the ease in which they can form and their mutagenic properties. Many active pharmaceutical ingredients, or APIs, contain nitrosatable amine groups and are therefore at risk of forming nitrosamine drug substance related impurities, or NDSRIs. Regulatory authorities require pharmaceutical manufacturers to risk assess all pharmaceutical products for the presence of N-nitrosamines, and where a risk is identified, develop methods to analyze them. Hi, I'm Tom Kershaw. I'm a scientist at Resolian in the Analytical Sciences department, specializing in trace analysis of nitrosamines in pharmaceutical products. A major pharmaceutical company requested the develop and validation of a quantitative method for the analysis of an NDSRI, the analyte, in a drug product for allergy relief. Several issues had to be overcome in this development, including the requirement for a sub 1 nanogram per mil quantification limit, a complex sample matrix, and a lack of a commercially available deuterated internal standard. The initial challenge was the separation of the analyte from the API itself. An analytical method had previously been developed at Resolian for quantitative analysis of the same nitrosamine in other drug products that contain the same API. This used an offline SPE cleanup step to remove the API from the sample solutions containing an aqueous diluent prior to the injection on a Z-helix column. Applying the same offline SPE process was found to be unsuitable due to the presence of cross-caramelized sodium in the allergy relief drug product. Upon contact with water, it rapidly swells to 4 to 8 times its original volume and therefore renders existing SPE cleanup process of aqueous solutions impossible to perform. Additional offline extractions were performed prior to the SPE step to attempt to remove the cross-caramelized sodium. These extractions made the overall preparation procedure complex and time-consuming. The lack of a commercially available deuterated internal standard meant that any matrix effects and impacts during extractions could not be compensated for. This led to significant negative impacts on analyte recovery, reproducibility, and sensitivity. The reverse C18 phase column retained the API and other excipients, while the analyte was poorly retained. Then, with this separation, the injection moved to the normal phase HILIC column, where the salts were retained and the analyte was not. This prevented the salts from enhancing or suppressing the analyte. So, comparing the offline SPE against the online SPE, we can see that the method is greatly improved by increased separation on the instrument. In conclusion, the online SPE enabled an effective and reproducible separation and sample cleanup, despite a lack of an internal standard and the complexity of the sample matrix. Using an online SPE mixed mode approach resulted in a successful GMP validation of the quantitative method for the analysis of the analyte in the allergy relief drug product, according to the ICH-Q2 guidelines, which also met the required criteria as directed by the sponsor, despite the difficulties faced. Thank you for your time. This was a quick overview of a method development here at Resolian. Please feel free to contact us or visit our website for more information.